Okay. Hi, greetings everyone. This is Mathew Cherian from United States. Nikolina Biskic will join me from Switzerland, and we will moderate this webinar. Today's webinar is on roadmap for drug product development and manufacturing of biologics. And the panelist is a highly experienced researcher and manager of this type of activity, Dr. Krishnan Sampath. He's Senior VP for CMC at Invetx. Dr. Sampath has extensive experience in development and manufacturing of biologics for humans and animals. He has well over 2 decades of experience. He has successfully supported regulatory approvals for several products. He has a suite of publications and patents. Dr. Sampath holds a PhD in pharmaceutical sciences. So the next slide. Krishnan, can we go to the next slide? Yes. Now, before Krishnan starts, I want to announce that all participants are welcome to ask questions. The way to ask questions is if you go to the bottom of the screen, there's a tab for Q&A. Type in your questions, and at the end of the presentation, Dr. Sampath will answer those questions one by one. So, Krishnan, it's all yours.
Thank you, Mathew. First of all, I want to thank Dr. Mathew Cherian and also the organizers of at FIP for inviting me to present this webinar. This is the second time I'm presenting at the webinar. The previous time was on quality by design approach of regarding how to do drug product development and manufacturing of biologics. Today's topic is going to be focused on the roadmap for drug product development and manufacturing of biologics, which is what and when to do, right? So that's actually the background for today's topic. So you see, yeah, okay, there we go. So much of what I'm going to be talking today is based on a recent perspective paper that myself and Bruce Kerwin, former colleague of mine, have published in the Journal of Pharmaceutical Sciences. The title of the article is similar to the webinar title. So the details of this talk can be actually reviewed by accessing this article. It's an open access free article, so I would encourage everyone to note it down and go through the article at your leisure. So as I mentioned, I'll start with at a high level what the universe of biologics looks like. What are the different modalities following common pathway of development? I see potential participants list. Based on the potential participants list, we have people from across the globe, in different time zones, and also different experience. There are undergrad, grad students, as well as regulators, scientists, a lot of folks attending. So I want to actually align and present a high level 30,000 foot level of universe of biologics, and then we'll start going to the different layers of what is the path from discovery to commercialization of biologics, and then the roadmap to development and manufacturing. That'll be the more detailed view of, okay, even still it's about 1,000 feet level of regarding how to actually develop the biologic drug product and development and manufacturing, and then we'll have a summary. I mean, it's a broad topic, so it is a 1 hour is not enough to cover all of this, and that's why it is important for I'll try to cover the key concepts during this webinar, and hopefully this article which I've provided here will answer a lot of detailed questions that you may have. So we'll start with the universe of biologics, right? What is a bio? What do you mean by biologics? A biologic drug, or what I'm going to keep calling it biologics, is a product that is produced from living organisms or contain components of living organisms. So uh, I mean, there are different modalities of biologics. Blood products and blood-based products have been used for centuries now. It's got advanced in the last 5 decades. And then I think from 1960s, 70s onwards, there is a use of this recombinant insulin. Then it evolved to cytokines and hormones like EPO and erythropoietin and GCSF. And it has evolved into antibodies, bispecifics, antibody drug conjugates. A whole class of antibody-based products have come out. And then the last decade, you see a lot of gene therapy and mRNA technologies that have come out, right? So now we have CAR T technologies as well. So we are actually in a very exciting phase when it comes to the biologics, right? So it's just amazing how the field has evolved just in the last 4 to 5 decades. So with that, I mean, I think while the biologics has actually evolved, the development and manufacturing knowledge of therapeutic biologic molecules has also evolved considerably. Just because of the background of the audience, I just want to ground ourselves. So traditionally, I mean, the small molecules like aspirin, right? It's a small 180 Dalton size. It's a small, but when you compare the biologics, right? When we go from the aspirin to insulin to human growth hormone to antibodies, the differences are in size, complexity, stability, and administration. So uh, uh, so the processes that you use for biologics is different from what you use for small molecules. We tend to use gentle processes. We can't use tableting, milling, suspensions. The traditional high mixing, those kind of practices or processes which are used for small molecules are not so much used for these biologics. You need to be gentle in the process because these biologics have structure because of the structural complexity which is required to maintain functionality. If you use stresses that can actually break apart these biologics, it won't function. So that's the reason why the knowledge of how to develop and manufacture of biologics is different from what is for the small molecules. And what has evolved, if you look at the right side of this, is the development of complex molecules and understanding as we go from early stages in the 1830s, from proteins discovered to now, it is just like tremendously, exponentially increased, right? So you go from insulin to the 1980s, where I talked about the cytokines, the antibodies, which came from the 80s to the 2000s and still going on. And then the recent last 10 years of gene therapy, cell therapy, we have come a very long way in understanding how to make these biologics. There are now more than 100 monoclonal antibodies that are approved as of a couple of years back, and monoclonal antibody based products as well. And also in the last decade, there is several of these gene and cell therapies. So there is tremendous evolution of knowledge that has come across, that has come about for development of these biologics. Now, the question still is we have a biologic. Here I show an antibody. We need to get from here to here, which is therapeutic products which serve patient needs, right? So ultimately, everyone is in this business to get life-saving, high-quality products to the patients. How do we do that without getting lost, right? As the number of biologics is evolving, how do we use this and the type of biologics is also evolving, the complexity is evolving. How do we take it from a discovery stage of a novel biologic to the end product, which is serves the patient needs, right? Patient centricity is what is important here. Um, Fortunately, the good news is while the biologics are becoming increasingly complex, the principles of development remain constant. So, what do I mean by that? So, you can have different cytokines, you have lipid nanoparticles, your adeno virus based products, the lentivirus based products, and CAR T cells. But if you think about it, everything uses similar guidances, right? There's some of the concepts like the quality target product profile, critical quality attributes, the way you develop the formulation, analytical methods, container closure, manufacturing, and stability studies are are similar, right? I mean, I think the principles are similar. So, and also there are several guidance documents. You say ICH guidances, FDA, EMA guidances, and of late there's a lot of industry consortium perspective papers that have come out, which helps folks developing these biologics to take it from discovery to the end product. There is also product specific documents that are how do you develop the gene therapy, how do you develop cell therapy based products. There is a lot of guidance documents available. But the concepts, as I mentioned, is the fundamental concepts remain constant. So that is why it is important for the undergrads, the graduate students, budding scientists who come into the field to understand the product, the background, but also be familiar with all these guidance documents and also literature, because the principles, the grassroots principles, and the foundation has to be solid for taking these products to the end. And for most part, if you got those principles right, you will be enabled to develop the product effectively. So now we are going from a higher level to a little bit more detail, right? So if you take the biologics product development, manufacturing, and patient, there are several factors that needs to be kept in mind. Similar to small molecules, biologics have their own unique aspects. You need to understand the molecule, the formulation that you prepare. The drug substance, just like API, we call it drug substance for biologics. The API the drug substance aspects needs to be understood. You need to understand process of making the drug product formulation. UFDF is ultrafiltration, diafiltration, and freeze thaw. You take the drug substance, you do the ultrafiltration, diafiltration into the formulation of your choice, and then we put it in drug substance containers, which goes through freeze thaw. And then before mixing the formulation step, and then filling either in vials or pre-filled syringes, and then there is a transport, and then storage, and then it's administered to patients. There is all these steps, right? So correspondingly, each of these aspects right, as I mentioned, formulation, the drug product process, the drug product stability, the device, what are the considerations for transportation, storage, and administration, what are the full finish operations aspects. If you're using a lyophilization, there are certain set of factors that needs to be used. For pre-filled syringes, there is a certain set of factors. What are the analytical methods used to monitor the drug product? And also, how do you ensure, in the end, how do you ensure the drug is administered to patients effectively? Because we talked about patient centricity, right? So how it's administered to patients effectively, all these come into play. Now, the reason I'm not going to go into each and every detail, that's not the purpose of the slide. It's to actually communicate or convey that there is a lot of these factors. But how do we actually address all these factors is to use a risk and stage-based quality by design approach that needs to be integrated to ensure patient safety and commercialization. Obviously, there are all these factors. Having a quality by design approach of what factors really matter for your product and also the process And having a risk-based approach is how we address all these factors, right? And so there's, as I mentioned, there's a separate webinar, and it's uploaded to YouTube, which I gave about 3 years on how on this concept of quality by design approach. But this is how to do it, is a quality by design approach. Now we'll talk about what and when to do it, right? So one thing which, just to ground everyone, right? And just because of the background of the audiences, we have different stages of drug development, right? Phases of development. There is a preclinical phase, there's phase one, phase two, part of phase two, which is called early clinical development. Then there is part of phase two and phase three, which is called late stage clinical development. Then you go into registration and launch and life cycle management. Now, one thing I want to reiterate is, the regulatory agencies, for most part, are willing to work with the sponsors of the drug in order to give the right guidance and direction for the drug manufacturers to develop. So there are product-specific meetings that could be done throughout the life cycle of the development, and the regulators provide that opportunity. Let's delve in a little bit into what these different phases are, right? So there's a preclinical phase. And this is when the drug comes from the discovery stage. We get to the CMC team. There are activities that are designed to gather information for filing an IND. And many of these activities are, hey, what is going to be your cell line? What does your process development look like? Or cell culture, purification, formulation, and analytics? How does your animal tox studies are going to look like? Clinical design for your phase one study. And then how are you going to manufacture the drug product by using GMP practices? And then IND writing and filing an IND application. This is actually preclinical to phase one. Then you have the phase one, phase two, and phase three clinical studies. Phase one, the purpose is to ensure safety of the drug in healthy volunteers. In some circumstances, actual patient, I mean, cancer or HIV patients are involved in this phase one studies. Then there is phase two, which is a dose finding or efficacy studies, where you're trying to find whether the drug is efficacious. You do the dose expansion, and also what is the right dose that you want to select for your phase three is selected using this phase two studies. And then the pivotal studies, where you go with your right dose and DP presentation, which is similar, which is for most part has to be the same as what we go for commercialization. So this is the pivotal study. Then right after that, by the time you get to registration or the biologics license application, it's called new drug application for small molecules. You start putting all these pieces together, and we'll talk about what does that entail for drug product development and manufacturing, right? And the review period is 10 months for FDA. For 90% of applications, there is priority review. For EMA, it's 210 days. And even after you launch, there is actually several activities that are ongoing: sales and marketing, post-market surveillance, phase four clinical trials are becoming common, and also the sponsors can switch. This is the time where they find that from a life cycle management perspective, they want to go to novel delivery devices. They can, many of the sponsors try to actually, of the drugs, they try these different modalities. So just to ground everyone, this is what we're talking about. This is the phases of development, right? Now, going on to the next slide, before that, I actually work for a company with respect to developing the biologics for animal health products. And for the when you look at the veterinary products, the preclinical data also provides serves as a pilot study for safety, PK, and efficacy. And for most part, you collect the information from here, which helps to leapfrog into the pivotal study based on this information. There's not necessarily a defined phase one and phase two, but for veterinary products as well, along with the human products, the same rigor and the same considerations apply. I wanted to actually convey that. Now, going on to the drug product and clinical development, these two paths are closely linked. So how do we? I mean, I think it's very important consideration. The deep drug product characteristics affect clinical design. What do I mean by that? So the concentration limits, right? If there is a viscosity and if you need to go to a higher concentration based on clinical needs, that can have an effect on the viscosity, and that can have an effect on the viscosity and aggregation of the molecule. Um, Insolubility at neutral pH. When you administer the drug into the body, the serum is right, the blood is at neutral pH. We need to look at whether the drug precipitates upon injection. Many of the drugs used, especially cell-based products, are needs to be stored at -20 and above. So there is actually limitation of when you develop the drug product, you need to be thinking about the limitation of available clinical sites with that -20 degree storage temperature. Is it possible to develop the drug product better? Low bioavailability for subcutaneous delivery, right? So the IV delivery, is it actually when you go sub-Q, do we have enough bioavailability for the clinic to happen? So these are considerations that need to be in mind while how does a clinical design impact the drug product characteristics? Then I think there's clinical needs, and the vice versa, right? And how the clinical needs that affect the drug product development, and also how the drug product development affects the clinic. In this case, let's look at the clinical needs that affect drug product development: the injection frequency, the IV delivery, blinding, and injection site reactions considerations, right? So do we need a drug which is slow release, or that specific formulation needs to be developed for that. For IV delivery, it requires development of formulation which confers stability upon dilution in IV bags. So compatibility studies need to be done. Microbial challenge studies for storage of IV bags greater than 4 hours, that needs to be done. If there is a placebo, there needs to be blinding. Colored solutions may need to be prepared for meeting the clinical needs. And also injection site reactions. Are those excipient related? Do we need to change the excipients based on that? So as you can see, there is very close interaction between the drug product development and clinical team, which is required for developing the right choice, right? And we'll talk about target product profile a little bit later, but this is the reason why the teams need to work hand in hand. And for that part, also marketing, right? Marketing, how you don't want to have a very complex delivery device which no patient knows how to use. So the marketing team needs to work with the product team in order to develop the right delivery mode, right? So those are the aspects which needs to be taken into consideration while developing the drug product. Let me see. It's not advancing. Next slide. Here we go. So what I'm going to be talking today is this roadmap, right? And I don't want everyone to start reading this. We'll talk about this in the subsequent papers, in the subsequent slides, and also it is conveyed in the paper. But what I want to actually talk about is how do we address all these factors? It needs to be a systematic approach, right? As you stage-based systematic approach as you go from preclinical to the late stage. And what needs to be kept in mind is as we go from preclinical to launch, there's increasing complexity of studies that needs to be done and increasing cost. But the way to offset is a stepwise, risk-based, stage-based approach, right? That's what we're going to talk about today. And the end goal is to maintain safety, efficacy, quality, and potency of the drug product during the development and commercialization. So that is the basis for providing this roadmap. And as I mentioned, this paper talks a whole lot about it, but I'm going to give you some of the flavor today, right? Again, a slide which I don't want to overwhelm the audience based on this, but the purpose of this is to convey that the reason why you want to do a stepwise and systematic approach to drug product development is there are all these challenges and issues that will come up during the early stage to late stage development. Say, for example, liquid formulations. You may have to develop a low or a high concentration formulation. Recently, polysorbate instability has been talked about causing particle issues in these liquid formulation of biologics. So there may be formulation and excipient related issues. Then there can be instabilities related to subparticle formation. There can be container closure issues, and also injection site or pain issues when you administer to the patients. And all of these challenges will may or issues may come up during as you go from the early stage to late stage, and that's why a systematic approach is required. To use a lyo formulation, just like in small molecules, there may be unwanted crystallization of excipients, which needs to be avoided. The lyo process may cause the protein to lose activity or the biologics to lose activity. If you have a long freeze drying cycle, then it makes the process not sustainable, right? It makes it very costly to make these products. Cracking of lyo cakes, and also heterogeneity of crystallinity, which causes reconstitution issue when you reconstitute a lyo cake. These are issues for lyo formulation development that needs to be kept in mind. And then we have devices. Now you take a liquid formulation, you put in a pre-filled syringes, then you have a whole set of factors to consider, right? The polysorbate level needs to be enough to protect the protein, but if it is too high, then the silicon oil in pre-filled syringes will start slowing off and cause issues. There is silicon oil layer in pre-filled syringes that may adversely impact a protein stability. So that needs to be addressed. You need to have enough silicon oil to have a good break loose and glide force in the pre-filled syringes. So there is this balance that you need to study. And then there's sterilization of the pre-filled syringes, which can cause changes in surface chemistry. So I just wanted to provide a flavor on the slide of these are, I mean, it's developing a biologics is not easy. A drug product is not easy. But if you being aware of these aspects, and this is important for the undergrads, grads, and the new scientists entering, is there's tremendous amount of resources of paper on these aspects, making sure that you have the fundamentals right, knowing all these issues, and actually using this, keeping this in the background that there are resources related to this, and while developing the drug and systematically addressing it is what is important. Okay. All right. Now, before we go into the next slide, the theme of the roadmap that I'm going to be talking about is to utilize a sustainable and systematic approach, right? So what do I mean by that? Follow the arrows. So when you start from early stage, you want to have material from the shake flask and early bioreactor studies. That material is used for early stage formulation development. And then you go into the tox batch, which serves as a lead lot for stability monitoring. Then after your formulation and the stability of the tox batch looks good, use that material to actually go into the phase one, use that knowledge to go into the phase one studies and make your batches, phase one and phase two batches. And based on the stability of the phase one and phase two batches, you determine, oh, is the stability good? If the stability is good, don't mess with it, right? But if there may be fine tuning or refinement that is needed. But if the stability is not good, you need to work on a pivotal or a commercial formulation, which is better. So then use that material to develop the formulation and the process further and then characterize and do DOE studies before you go into the engineering and pivotal batches. And then after Based on that, once the engineering and the pivotal batches look good, then you take those and go into your PPQ or validation batches. So the theme here is systematic approach using representative material and not actually front load a lot of activities, except if it's for biosimilars. Having a stage-based and a risk-based approach will help a lot um, to ensure that the safety, efficacy, and quality and the potency of the drug is maintained. I also talk about control strategies here. So you see that these efforts, right? The process control strategies, the CQA, process and raw material specifications. These build on systematically as you go from early stage to late stage. There is a progressive increase in these activities, right? So that's why it is important. As we go to from early to late stage, there are several tools which ensures that the process and the product is suitable, right? So you want to use this approach. All right. Now let's start delving into what are these different stages of development, right? Biologics development. So first one is the early stage, and the focus here is speed to clinic. Okay, so that's why I show several people running here, because you want to actually, once the drug is discovered, you want to actually get it to the patients and see whether it's safe and efficacious. That's the purpose of this early stage. So so the CMC activities during the stage is for first clinical dose to be administered to patients. So the CMC team has to start a little bit earlier, even during the research and the preclinical stage, to ensure that they are thinking about this, right? The goals here are what is the to develop a QTPP, and we'll talk about it. This is the quality target product profile. How is it actually, how to build a quality target product profile based on clinical design, analytical targets, and commercial targets. You need to have a robust, flexible formulation that takes you through the phase 1 and phase 2. So what do I mean by that? You want typically a drug product which is stable for 2 to 3 years, which can be frozen, but also nowadays most map-based products can also be stored in liquid, 2 to 8 degrees Celsius. Shipping it frozen is easiest, especially for cell and gene therapy and antibody drug conjugates. But shipping during the 2 to 8C can also be managed for many of the stable antibody-based products. Delivery is usually IV, but also sub-Q can be introduced. Typically, a container closure at this stage is a vial or a vial with a stopper. Pre-filled syringes are rarely used. And then you want to start collecting product quality data. So you want to start conservative and start collecting product quality data and particle data to understand your control strategy. So that is from a development viewpoint, right? And then we make a tox lot, which can also be used as a lead lot for expiry dating of the GMP lot before going into a clinical DP manufacturing using a CMO of choice or internally. So the considerations here at this stage is speed to clinic, right? If the sponsors have a platform formulation product and process, they can use that. But also there is industry-wide knowledge which can also be used to ensure the speed to clinic. And this platform knowledge can be based on known product instabilities, quickly assessing whether there are major instabilities of these products. Then a specific formulation or product needs to be developed, but there are no major hotspots in the molecule. Then it lends itself better for the platform formulation. Then what are the process impurities, pharmacy requirements, analytical targets, and regulatory requirements? All these goes into whether a platform formulation can be utilized. And then you also want to think about a little bit further is when you're manufacturing this drug product, what is the timeline of when these analytics needs to be done? What type of analytics needs to be done? And also at this stage, do we want to use a latest and greatest isolated technology or RABs? How does a product, what is the specific requirements for your product? And then a release and stability testing site is a different from the CDMO manufacturing it. These are different considerations at this stage. Now, I mentioned quality target product profile, and this actually has elements of target product profile. This is fundamental for developing your drug product development and manufacturing, right? So you start with understanding, working with your clinical counterparts. What is a clinical indication? What is your target population, dosage, administration form, marketing considerations? You don't need to have a final picture, but you need to have a high level picture of this target product profile. And then we understand based on the CQAs of the biologic, which can affect the safety, potency, strength, and quality. You need most of the sponsors develop a draft quality target product profile. So what does this mean is based on the patient needs or patient convenience, you have we have what does a formulation, what is a good suitable formulation, how you want to deliver it, what is the dosage strength and clinical use, what is a good container closure to use it. You don't want it in plastic container in large bottles. You want it to be easy for the healthcare professional to administer to the patients. And what is your stability profile and quality profile should look like? So that is what I meant by quality target product profile. And as I mentioned, you don't have to have a final version. These two documents, the target product profile and the QTPP, continuously evolve during the development. You need to have a high level blueprint. I call this as a blueprint for how you want to develop the drug product and as well as the process, right? So you need that. And I give you an example here. This is an example of a QTPP, right? During the early stages, right? For phase 1 and phase 2, you develop initial QTPP and then refine it later, and then it's refined again by the time it goes to pivotal. So you see a different several times refinement that happens. It's a living document. Why is it important? Is here is an example of a phase 1, phase 2, right? A phase 1, phase 2 formulation or a drug product is speed to clinic. So here is an indication for arthritis, and it's an IgG1 antibody. Intravenous is the most common way to start it. You have a low concentration, 25 to 50 mg per ml, relatively low concentration. When you talk about antibody-based products, it's in glass vials and stoppers. Then you have limits, typical limits, 5% for aggregates and 5% for fragments. But by the time you get to phase 3, you have product process understanding, and also you have multiple lots on stability. And then based on the QTPP, which is advanced, right? You start thinking about, oh, it's for arthritis. It may be for at-home administration. So let's make it easy by making it a subcutaneous administration so that patients can take the prefilled syringes and administer at home. So that's how the Humira, Enbrel, all these different products which have come out have evolved from is by thinking about what does a patient need. And that typically happens during the pivotal stage, prior to pivotal studies, right? And then in order to enable the sub-Q administration, you have to go higher up in protein concentration, use a glass prefilled syringe. But also you start actually based on the stability profile and patient exposure, you start actually tightening your specs or thinking about how do you actually have a better product quality. So here's an example of a blueprint or a roadmap of how your development target should be. And the development target typically comes from the quality target product profile. Okay. All right. So we completed talking about the early stage. The early stage, the focus is fast to market, use platform approaches where possible, and make the file the IND and get the product into the patients to evaluate safety. Now comes the mid stage, which is a development geared towards defining a robust commercial drug product. So the mid stage is you already the phase 1 formulation is developed, phase 1, phase 2 that is ongoing. then we start thinking about, based on evolving phase 1 and phase 2 data, is what now I have, like, I mean, I have we have the phase 1 formulation drug product, we have stability, we have made multiple batches. What do we need to think about to make this, if it is going into the pivotal, how to make it happen, right? So the CMC preparation for pivotal clinical stages start during the phase 2. So CMC always goes first, right? The clinical team may start talking, I mean, may not be there for the clinical pivotal clinical studies, but the CMC team, based on the evolving clinical data, and that's why they need to talk to the clinical team, based on the evolving clinical data, needs to start thinking about, oh, I need to start designing for the pivotal clinical stage. And that's the goal of this stage, the mid stage, or the during it happens during the phase 2 and goes into the phase 3 stage. You refine the QTPP we talk about. So going from a vial to a pre-filled syringe, you evolve the analytical methods based on the critical quality attributes. And sometimes, I mean, most times you go from you develop the pivotal formulation, which is also going to be used in commercial setting, right? Um, so it can be a liquid, it can be a lyo, as shown here, these vials, it can be a pre-filled syringe. You start thinking about how to make this product robust enough so that it can go through the pivotal phase, but also commercial phase, right? Then, then the CMC folks, the drug product scientists start thinking about phase 3 support studies. Okay, for this to be administered across the globe, the phase 3 sites or pivotal sites can be across the globe. How do we actually start thinking about it? We need to think about, okay, it can be administered in Africa, it can be administered in Asia. So we need to start thinking about what are the what are the consumables available there? What is the temperature over there? And then we have to do corresponding microbial challenge studies so that the length of the administration can be supported. There's also device compatibility and process development of these DP production that needs to be taken into account. So now we get into a next layer of details that needs to go on. And there is, as I mentioned, there are a lot of these considerations. So for QTPP, you need to think about commercial population, phase 3 population, right? For developing the phase 3 formulation, you need to start seeing, okay, how does a molecule degrade? What are the stability issues? What is a pivotal clinical dose? Is the marketing group planning on making it available for at-home administration through a pre-filled syringe and an auto-injector? So all those considerations, and this is why, I mean, I think it is interesting because the challenge is the clinical team is still in phase 2, but the drug product scientist needs to be almost have a crystal ball and start work with the clinical group as well as the commercial group in order to start planning for the pivotal stage and the late stage. Then there is clinical study considerations. That is, you're going to administer this drug to across the globe. So what are those considerations? Make the pharmacy manual crystal clear. If a placebo is going to be used in phase 3, how are you going to blind it so that what is the blinding strategy so that the healthcare professional or the patients do not easily know it's a placebo versus a vial? If you're using a pre-filled syringe, what are the primary container interactions that need to be addressed? And also, since this is the pivotal phase and also the commercial phase manufacturing, people have to think about studying the unit operations for the drug product process development more thoroughly, right? So that is what happens. So the bulk of a lot of this commercial and pivotal formulation development happens during the mid to late phase 2 into early phase 3. So these are the considerations. And to simplify this, right? So the formulation scientist needs to be thinking about 4 aspects. You need to have a drug which is manufacturable, has good stability. You want to have 2 to 3 years shelf life, not just in the US or UK or EU, but also for global expansion in India, China, Africa, Australia, we need to think about whether it's going to be stable there. How being patient centric, how to make this easy for and safe for the patients. The formulation scientist needs to be thinking about it, and also marketable. How to lower the cost and make it easy for patients, right? So that's these are the considerations that need to take place while developing the drug product formulation. And as I mentioned, I showed you previously, there's a long list of challenges. Some of the challenges is again reiterated here. The formulation scientists and the drug product scientists need to think about how to actually address this during this phase stage, right? Um, there may be particles in biologics, vials, and prefilled syringes. How do you effectively address that? There may be deamidation, oxidation, which actually makes the molecule less active. How do you control the CQAs for that? How to have a robust unit operations? So here, I mean, a typical list of unit operations can be drug substance thaw, pooling and formulation, bioburden reduction, filtration, then sterile filtration, filling, and then capping, right? But each of these unit operation needs to be studied during this mid stage in order to come up with a robust process. Um, what is the optimal formulation space for your pivotal and commercial formulation? What is the right? How do you actually make it injectable to the patients? If you're using a pre-filled syringe, how do you reduce the viscosity? And how do you make it easy to go into an auto injector, which may be used for pivotal or the late stage? So these are challenges or issues that the drug product scientists need to be thinking about at this mid stage of development. All right, I think we are making progress. Um, the last couple of stages is the late stage, right? Now, when you come to the late stage, is gearing towards pivotal study support and process characterization. So remember, the CMC team is always needs to be one step ahead of what the clinical team does. So by the time we get to phase three pivotal, you already finalized your pivotal formulation, you've made your pivotal batches, and now the drug product scientists need to be really thinking about, okay, what steps do I need to validate the process, right? So again, the QTPP goes through refinement. um, you have to establish at this stage, you have to establish the pivotal study and commercial manufacturing site. It is important to ensure alignment of DP production with internal or a CDMO process. You ideally want to have the same site, CDMO site, for making both the pivotal as well as the commercial material, because there may be different product profiles which you may have, and so compatibility becomes an issue. So you want to select the pivotal study and the commercial study, commercial manufacturing site to be similar, and that starts happening during right before the pivotal studies start, right? um, when we want to do engineering studies and get ready to make these batches for the pivotal release. You want to do delivery device compatibility studies. If you're using a pre-filled syringe or an IV bag, now at this stage, because of the global sites, you may have different IV bags and lines to study. You need to understand what these phase three sites use, and you need to accommodate that during this stage. Want to refine the process control strategy and then make the pivotal batches. Um, these are all the pivotal leading to the pivotal batch manufacturing, which goes into the pivotal studies. But during this late stage, while the pivotal studies are ongoing, the drug product scientists need to keep an eye on how the product stability looks like and start planning for their next stage, which is the process validation stage. So there are a couple of activities: process risk assessment and process characterization studies that need to start at this stage. um, I'll briefly talk about this in the next couple of slides. um, I mentioned the life cycle of process validation involves process design, commercial process qualification and continued process verification. So by the time you get to the late stage, you're done with process design. So all the risk product development, process characterization activities need to be complete. This usually is completed between the mid to late stage of pivotal clinical studies, right? And then you need to start getting ready for this commercial process qualification, and then getting ready for the continued process verification. This is based on the guidance for industry process validation from the FDA. But this is what we have been talking about so far. The development and characterization is still stage one. It needs to be, then the process needs to be qualified and then verified. So there are good papers talking about the process characterization activities. So mid-stage, you lock in the process, and then during the late stage, you need to characterize the process. What do we mean by characterization? So based on the product knowledge, you have the critical quality attributes identified. So the critical quality attributes identification forms the basis of process characterization studies, right? So once you identify the CQAs and develop the methods for it, then you do a process characterization. You take the process, you look at what is called the normal operating range, defining what needs to be the normal operating range, but also proven and acceptable range. What are the full range of process parameters to evaluate to get to this qualification stage? Does the process control the critical quality attributes, and is your product stable? That's what you answer during this late stage. So as I mentioned, there are you can note down these references. There are good references about it. If you do a literature search, you can read how quality by design approach is used. But a drug product process risk assessment should always be used. So once a CQA is identified, you do a risk assessment, then come up with what is your process range that you want to study, and then you study it, right? So you typically process characterization is then done prior to PPQ. So some examples, and again, the goal here is not to talk about how to do it, but what to do it, but I give you a flavor, right? So taking the key process and formulation parameters, it is possible to combine it all in single studies. You can combine formulation and process, taking the worst case considerations. You can say, for example, your product is susceptible to freeze thaw, combining that with the formulation variables, having a headspace for oxidation, and then transport being a stress. All this could be studied through a properly designed DOE studies. So that's what you do during the late stage, is how effectively can you combine the high risk parameters and study that systematically. Now the last stage, and I want to give 5 minutes for questions and answers. So I'll go through this very relatively quickly. Is last stage is primarily is called the PPQ stage, process performance qualification stage, and is highlighted by doing your process validation batches as per the regulatory guidance, and then preparing the BLA, right? So and the goal here is to prepare for market launch. Again, this is towards the end of the pivotal studies, but the drug product scientists are already need to be thinking about commercial launch. So there is a whole set of activities related to that. And the article that I cited will go through it. You want to finalize your CQAs, you want to finalize your manufacturing. And then the manufacturing risk assessment needs to be done. There needs to be risk mitigation studies for manufacturing, which needs to be performed. Then you want to have a process validation plan and a final control strategy. And then you actually have a commercial manufacturing site identified. And then we go into performing the process validation batches, setting those on stability. And then you have um, other validation studies, right? You have a continuous, you have transport validation study, extractable, leachable studies. All these are performed at this stage. then you have to have a continuous process verification program in place, and then start writing your BLA and preparing for the pre-approval inspection, right? So that's here at this stage, you are pretty much finalizing at this stage of process validation. You're in the mode of addressing any residual risk and preparing how this is going to be launched commercially. So during the process characterization stage, the focus is to mitigate the process risk and finalize your normal operating range and the proven acceptable range. But for process performance qualification stage, now by this time when we get here, right, the PPQ stage, you should be able to have what is your critical product, process parameters, key process parameters, have those defined. Then you have to do a manufacturing risk assessment, whether there is a facility fit of this process in the final manufacturing site. And then you define your control strategy. So the focus at the process validation stage is these three aspects. And it's quite similar to how small molecules are done. It's also the same concepts are used for biologics process performance qualification as well. Last slide before I go into the conclusions is, so at this stage, as I mentioned, you will have your final formulation, process, control strategy, and packaging. These batches, the primary stability batches, are from the pivotal and registrational batches, which are ongoing. This will give you the shelf life, define the shelf life. And by the time of filing, you typically have 12 or 18 months of the stability. Then the CMC team executes the drug substance validation, followed by the drug product validation, and then the delivery system validation. And you want to systematically put these batches on stability. And by the time of the filing, you will have only typically 3 or 6 months, but hopefully you'll use the registrational batches, the 12 or 18 months from the registrational batches to set the shelf life or the expiry dating for your product, right? So as you can see, these validation exactly overlaps during the phase 3 clinical trial. So by the time the phase 3 clinical data evolves, you need to be done with your validation so that you can file. The clinical trial completion is done and you have enough time to put your BLA together and submit it. So the CMC team or the drug product team needs to be thinking 2 steps or 1 or 2 steps ahead on how to devise it. You don't want to go too fast because sometimes that can be phase 3 failure, but you need to plan for these majority of this validation as soon as you, as a clinical team, signals that there is positive data, you want to execute on these validation batches and have it ready for the BLA filing. And for accelerated development, sponsors have worked with the regulatory agencies to determine appropriate timing of these validation batches. Some of these validation batches can be, some of the pivotal batches can be used for the filing, and the validation batches are done during the review period of your BLA. And then the stability of these batches can also be discussed with the regulatory agencies for accelerated development. And this is a continuous process. Last but not the least, this whole process validation is a continuous process. Anytime you go from process design, process qualification to CPV, if you make changes, then it goes back to process design stage. How big of a change? If it's a major change, right, it needs to go back to stage 1. If it's minor change, then you do a risk assessment and discuss with your regulator, hey, the product impact is low, can we proceed to the next stage, right? So that this FDA slide is very important for everyone to remember. All right. So I think I want to give a few minutes for the questions, but hopefully I actually emphasize the need for streamlined QBD-based approach using a QTPP and a stage-based approach for development. And if you want to use novel approaches like artificial intelligence or machine learning, that needs to be incorporated during your phase 1, early stage and mid stage, so that you have enough time to validate that approach. And the end goal is patient centricity. So I'll stop there. And I want to actually answer a few questions that are in the Q&A panel. I actually, yeah.
Thank you. This has been a very comprehensive, thorough presentation, and you took a lot of pains in preparing this for us. Thank you very much. I'm going to ask Nikolina, she's my co-moderator, to help you with the Q&A. She can read out the question and you can answer if you don't mind. So Nikolina, please go ahead.
Hi, everyone. Yes, thank you, Mathew. I just want to mention to everyone that I saw some questions asking for certain journal links and so on, and for slides. So in case you want anything from that, please feel free to contact Krishnan at this email stated on the slide. So let me start with the questions. The first question reads, how can we empower a developing country to develop and manufacture drugs and biologics, and what could be the possible challenges and opportunities in those countries?
Yeah, thanks. Yeah, I know that's a broad question, right? And it's an excellent question. From a developing country perspective, as I mentioned, many of these concepts are similar. The principles, the fundamental principles are similar, right? So I would suggest, I mean, being familiar with all these guidance documents and thinking about from a developing country perspective what your local regulators want. Um, the product and the process science is across the board, across the globe, has to be similar. The end goal is to have safe, efficacious, high quality, affordable drug. But how do you actually develop the challenges may be to actually go maneuver through the local regulators and also have think about the supply chain and the pharmacies and clinical sites in your specific country. That's where the logistics is the major challenge. How do we actually ensure that the product which is high quality is delivered to the right patient? So the logistics is the one which I would say is working with the regulators there, finding a pathway is the main challenge. But I want to reiterate the concepts, the fundamental principles across the board has to be is quite similar. Hopefully that answered the question.
Great, thank you very much for your answer. So I will go ahead. One of our attendees asks, is the drug product development roadmap same for small molecules, biologics, biosimilars? If not, what are the main differences?
Yeah, thanks for the question. So this one, the roadmap is, as I mentioned, the pathway of development, the clinical stages are quite similar. But as I mentioned, the complexity related to biologics, there are subtle aspects that needs to be taken into account. Um, the fundamental principles may be somewhat similar to the small molecules, but biologics have their own. There needs to be the processes, the formulation aspects are unique to the biologics, and that's where the challenges and issues are different than the small molecules. Which that's why I think for biologics and biosimilars across the board, those are quite similar, and that's why we created this roadmap. And I would highly encourage the readers to go to this to develop that appreciation or awareness of what these unique subtle aspects are compared to small molecule development.
Great, thank you very much for that. There is also a question regarding artificial intelligence use. So the attendee asks, how are developers strategizing to integrate advancements in AI into the development process of biologics, such as optimizing drug design, accelerating drug discovery, and enhancing manufacturing processes?
Yeah. And as you can realize, that's a broad topic which requires its own session, but it's a wonderful question, right? So we live in a world of AI, and what people have started using is this AI ML techniques in order to a lot of understanding of biologics has come in. So how do we use already evolved data? You train the system using this large language modules, and you actually use data mining and learning aspects for coming up with better drug candidates, right? Better map products. process is the same. People don't need to do thousands of formulations anymore. Taking a particular molecule and feeding the right data set, you can come up with these processes very quickly. So across the board, the same AI concepts can be used for drug discovery, formulation, process development, and also how to make it device compatible. You need the right data sets to feed into it, but there is a lot of these bigger companies which are already utilizing these platforms.
Great, thank you very much for that. So I have another question. What specific challenges do developers encounter when developing non-platform therapies compared to platform biologics, considering factors such as manufacturing complexity, scalability, regulatory hurdles, etc.?
Yeah. Fortunately, I mean, I think here is what I would say is when antibodies were first manufactured, people used the same route as what was used for actually developing the cytokines and commercializing. So there was no science which was unique. So people applied the same concepts, guidance documents, which were applicable to cytokines. And then when you look at the gene therapy, there is again the same concepts for mAbs are being used for the gene therapy for formulation and product development. Yes, there are unique things. Gene therapy and cell therapy products needs to be the supply chain and the storage conditions are different from mAbs. Those are not as stable as mAb products. But as you can see, I mean, the overall guidance and the pathway is built on from previous molecule or previous experiences. Um, and the regulators are willing to work with the sponsors in order to maneuver through this because I mean they just like the sponsors, the regulators are also trying to get these life-saving medicines to the patients as long as it's safe, efficacious, and high quality, right? So they I've seen a lot of sponsors work with the regulators hand in hand in order to actually come up with these newer therapies. The FDA has, for example, created a new division itself for addressing the cell and gene therapy products, right? So, but the short answer is you don't need to reinvent the wheel. Use the concepts or guidance documents already employed for successful biologic therapeutic products.
Thank you very much for your answer. There is also one question directed specifically to presentation 1, slide 16. The question reads, what minimum stability data is demonstrated before you feel comfortable to use the product in phase 1 studies?
Typically, I mean, that's why the tox lot is typically put on stability, and you want to have at least 3 months of a tox lot data in order to have a comfort level. If you have the 3 months of data from the lead lot or the tox lot, and you don't have a lot of formulation changes for phase 1, for phase 1 studies, 1 month of data is typically provided to the regulators. But if we do change, making major process changes, hopefully the formulation shouldn't change. Major process changes, you would ideally want to have 3 months of data, and that can come from the tox lot if the formulation and process is similar.
Great, thank you very much. We also have another question related to strategies. So what strategies are in place to anticipate and adapt to potential future challenges, advancements, or regulatory changes in the landscape of drug trials for the development?
So, yeah, I mean, I think I don't have the perfect answer to this question because it's a broad question, right? Um, but I would say, I mean, I think look at CAR T therapies, right? I mean, uh, 7 years back is how are we going to get the cells from different patients in order to actually these autologous aspects? How do we get these cell therapies to the right patients, right? The supply chain has worked it out itself. Uh, and again, reiterating how we did it was applying the fundamental key aspects of what was done before. Now, I mean, I think doing cell therapies, people have figured out the whole supply chain, and also how do we the leukophoresis, taking the product, purifying it into a drug product. All this has been relatively worked out to a scale that people are talking about large scale manufacturing, establishing large scale antibody like huge centers for making these cell therapy products. So there will be challenges as it evolves. There will be different world populations that need to be looked at. But again, it has to be a slow, systematic approach reflected through these guidance documents. That's actually, I go back to that answer each time.
Great. Yeah, thank you very much for that. So those are all content-specific questions, but of course, if any of the attendees have any more questions, feel free to contact Krishnan at this email. Mathew, I give the floor back to you.
Okay, so thank you very much, Sampath. This has been an awesome presentation, so informative, full of details. This will be valued by everyone. I want to state that a recording of this presentation with the slides will be available on the FIP website in a couple of days. It will be there for at least a year, and we will invite you to go take a look. I want to announce that our next World Congress of Pharmacy and Pharmaceutical Sciences will be held during the first week in September in Cape Town, South Africa. All of you are invited to attend. And by the way, South Africa is a very beautiful country to visit as well. So I hope you can come and join us for the World Congress in September. I want to thank Krishnan again for the fine presentation and thank all the attendees who took time to attend this session. Thank you very much.
Perfect. Thanks for the opportunity. And as mentioned, if you have any questions, please reach out to the email that was provided before. And thanks for the opportunity again.